Welcome to Nikolai's genetics lessons, and today we are going to talk about Manhattan plot, how to understand results, and this is going to be my sixth video. I highly recommend you to watch my previous videos, but nevertheless you also can start from this video if you have basic understanding of Manhattan plots. To understand fine mapping, you have to transition from thinking like statistician to thinking like molecular biologist. Fine mapping is a process of sorting through the statistical crowd to pinpoint the exact causal variance. So you got these hits, these single nucleotide polymorphisms in the genome that is highly associated with certain disease of your study. And the most straightforward scenario is when a highly associated SNPs directly change the genetic code inside the exon, a protein coding region. For example, take a look at this picture. This is how we obtain our results. So we take one group of people who has same disease in common. Then we take a general public which doesn't have this disease. Then we analyze whole genomes of this group of people, and this is going to be results of single nucleotide polymorphisms in their genomes, and here is the same single nucleotide polymorphisms would be, which is true for a group of people which doesn't have this disease. And that also means that here we should find certain pattern of single nucleotide polymorphisms, which is only can be found in this group and cannot be found in this group. This is how we find those hits. To complicate things, we also can find the same single nucleotide polymorphisms in this group of people, but in much smaller quantity, in much smaller frequency. Now take a look at this table, and this is codon table. We have 4 nucleotides which can form 64 codons, which specifies 20 amino acids. 1 also specify start codon, and 3 specify stop codon. Sometimes we can get silent mutation when third position in codon may change, but as you see, it doesn't matter to which nucleotide it's going to change, it still specify the same amino acid. But in this example, we see that first 2 positions are the same, but the last position, when it change, for example, C change to A, that means that phenylalanine would change to leucine. And of course, change in the first position and second position always means that this is going to be a change of amino acid. But I have to say that we are going to be very lucky if we find a hit, and we will find that it leads to change in amino acid, and such change of the amino acid may disrupt, for example, active site of the protein. So now take a look at this picture, and what you see here, this is protein, say some enzyme that speeds up certain biochemical reactions, and with blue color you see here binding site, and here is a molecule, and with red color you see catalytic site which speed up reaction. And if this amino acid change due to mutation in the last position would happen anywhere here, this is scaffold, usually it doesn't change protein or enzyme function seriously, but if such mutation is going to happen in active site or catalytic site, this may affect how effective this reaction runs, or maybe it's not going to happen at all. So we have a disease which is caused by single nucleotide mutation or single nucleotide polymorphism, which we have found in certain location. This type of mutations we call missense mutations. If a SNP changes the codon, so it translates into different amino acid, it can alter 3D folding of the resulting protein, disrupt an active site, or cause the protein to degrade prematurely. Another type of mutation we call nonsense mutation, when change in any position of the codon may turn it into stop codon. In this case, there can be a break in splice site, resulting in truncated non-functional protein. And as I said earlier, coding mutations are easiest to understand, but they account for less than 10% of the significant GWAS hits. The vast majority of the disease-associated variants lie in the non-coding dark matter of the genome. Maybe many of you know that only 2% in our genome account for coding proteins, or we call this genes, which specify certain proteins. And within gene, there are exons and introns. And exons account only for about 10, 15, or 20%. And within a gene, introns, which non-coding sequence, account for up to 80, 90%. So as you see, in most cases, such mutations, just statistically, is going to happen in non-coding regions. But how they affect our health? So I'm talking about epigenomics, disrupting tissue-specific switches. If causal SNP is non-coding, it usually acts as volume knob rather than a structural blueprint. It alters how much of a protein is made or where it is turned on. It does this by disrupting regulatory elements like promoters and enhancers. Now I want you to take a look at this picture, which describes how epigenetics works. You see there are epigenetic factors which attach to histones tails, which specify accessibility of the transcription and translation mechanisms to DNA. If SNP causes the DNA to tightly wrap back around the histone proteins, cellular machinery can no longer access that segment of the genome. And this also may affect how certain gene would be expressed. Next mechanism, which I want to discuss, imagine that here we have a gene. In the upstream region, we have a transcription factor binding site, and a single nucleotide change can completely obliterate the binding site for crucial transcription factor. If the factor cannot bind, the downstream gene is not activated. So this is completely different mechanism, but this is type of regulatory mechanism which may affect how gene expressed, or probably it won't be expressed at all. You also have to pay attention to tissue specificity. This is ultimate smoking gun. If you are studying GWAS for Crohn's disease and inflammatory bowel disease, a causal SNP should ideally sit within an enhancer that is open and active only in immune cells like T cells or gut epithelial tissue. If the region is only active in brain tissue, that SNP is most likely just a statistical passenger. And now I want to talk about also three-dimensional chromosome structure. As you see, it is very complex, and sometimes sites which are separated by thousands and sometimes by millions of base pairs, when chromosome in a tight formation, those sites can be just next to each other, so can affect each other, even those between them can be thousands, even millions base pairs. Perhaps the most deceptive scenario in GWAS is when a massive peak forms in the gene desert, a vast stretch of the DNA with no known protein coding genes for hundreds or for thousands of base pairs. Initially, this hits baffled scientists. However, we know now that the genome is highly folded inside the nucleus, and enhancer located millions of base pairs away can physically bend, form a loop, and physically touch promoter or target gene. So how fine mapping solve it? Researchers use spatial molecular techniques like high-C or micro-C, which map 3D chromosomal architecture. If non-coding causal variant in gene desert sits inside the enhancer, 3D looping data can trace exactly which gene promoter is physically contacts this gene. So the whole purpose of this video was to show you that hits that we getting in GWAS is not necessarily have to be within coding sequence within genes. As you see, you have to get a broader look, and such single nucleotide polymorphism may also happen in different places of our genome, in introns, in intergenic space, and in the gene deserts. One more time, less than 10% of all single nucleotide polymorphisms account for change of the amino acid sequence. And this is all for today. Subscribe and see you in the next video. Goodbye.